Hello, everyone. My name is Michelle St. John, and I am a rising fifth year in the Integrative Biology Department at the University of California, Berkeley. And I am very excited to tell you all about my recent research on the intersection of the evolution of novelty and the formation of reproductive isolation in the pupfish system. So novelty can be defined as any new behavior, trait, or new combinations of old traits and behaviors that allow an organism to occupy a new ecological niche. And I think understanding the evolutionary origins of novelty is interesting because they are not particularly well addressed by Darwin's theory of evolution, nor are they well incorporated into the modern synthesis. And I think one way to address this gap is by investigating the relationship between novelty and the formation of reproductive isolation. So there are currently a few examples illustrating this potential relationship. So first is an example of a positive relationship between novelty and reproductive isolation, where the evolution of novel migratory behaviors in this European blackcap produced assortative mating. However, there are empirical examples to the contrary. So for example, the study in corals found that populations with increased levels of gene flow also had high levels of evolutionary novelty. And then finally, there are verbal models that describe the potential relationship, and they argue that selective pressures from the occupation of a particular ecological niche can directly affect things like reproductive isolation. So all of these examples agree that there may be a relationship between novelty and reproductive isolation, but they do not reach a consensus on what that relationship should be, which brings me to today's topic and my presentation. So I'm going to start with an overview of my pupfish system. I'm going to move on to describing 3 studies that I've performed to help tease apart the relationship between novelty and RI. And then by the end of the talk, hopefully I will have convinced you that there is a strong positive relationship between novelty and reproductive isolation in pupfish. So this is Cyprinodon variegatus, which is also known as the generalist pupfish, and they can be found across the Atlantic coast of North America, throughout the Caribbean, and even into South America. These fish eat typical fish things, so algae, detritus, and insects. However, if we zoom into San Salvador Island in the Bahamas, we will actually find a young adaptive radiation of pupfishes, which include 2 additional specialist species, the scale-eating pupfish and the snail-eating pupfish. So for today, I'm going to focus on the snail eater, but if you want to read more about scale eating, please check out one of my recent publications, and the citation is in that bottom right corner. So about 30% of the snail-eating pupfish's diet is snails, and we hypothesize that the snail eaters can consume snails due to this novel nasal protrusion seen here. And this is a bony extension of the maxilla. Snail-eating pupfish also exhibit a novel behavior of oral shelling. So while most Staurophagus fish will consume snails by crushing that outer shell, pupfish will actually wedge the snail out of the shell to consume it. So this is a great example of novelty because it spans the biological levels of behavior, morphology, and ecology. So I'd like to jump into some of my methods and results, starting with my first study. And the question that I investigated here is, what is the relationship between aggression and novelty? And I was interested in this relationship because based on the feeding video, we might hypothesize that aggression is important for not just scale eating, but also snail feeding in pupfish. It's also interesting because aggression can be used during mate acquisition. Pupfish partake in a liking system, so males often have to compete directly for females. So to investigate aggression, I measured it using 5 different groups of pupfish, and I utilized these mirror assays seen in the video. So we used the number of attacks as a proxy for aggression. We also looked at differential expression and aggression-related pathways between specialist and generalist pupfish. So what we found was that snail eaters and scale eaters had increased number of attacks compared to specifically generalist outgroups. Furthermore, this pattern seems to be primarily driven by male scale eaters and female snail eaters. We also did find evidence of differentially expressed genes between both specialists and generalists in aggression-related pathways. And interestingly, these genes were also annotated as having effects on things like androgen production and maternal care behaviors. So my takeaway from this first project is that aggression is related to novelty in the pupfish system. And from our transcriptomic data, this was the kind of first hint that there might be a connection between novelty and reproductive isolation in the system. So next, I'd like to tell you about a study where I investigated the oral shelling performance between purebred pupfish species and snail-eating hybrids. And the goal of the study was to help tease apart whether poor hybrid feeding performance was a form of isolation in the system. So to investigate this, we measured the proportion of snails consumed by pupfish over a 48-hour period and used that data as a proxy for snail eating performance. And I want to take a moment to recognize that much of this work was done by a fantastic undergraduate researcher named Christy Dixon, and it would not have been possible without her. So what we found from the study is that snail eaters and their hybrids consume similar numbers of snails despite the variation in nasal protrusion size. Furthermore, the generalist pupfish also consumes statistically similar numbers of snails, and this species doesn't really have a nose. So overall, what we think is happening is that snail consumption may be behaviorally mediated, but does not appear to be a source of isolation between snail eaters and generalists. All right, lastly, I would like to talk to you about some preliminary data I have on estimating reproductive isolation in the system. So for some background information, specialist pupfish only make up about 6% of the entire pupfish population. So if they are mating randomly, we might expect a lot of hybridization with generalists. However, when we look at ancestry from genetic data, we can see that snail eaters actually have far less variation than generalists, suggesting that they might have higher levels of reproductive isolation. Furthermore, a few previous studies did kind of look at conspecific preferences and did see that all three species seem to prefer their own mating with their own species. However, they only focus on prezygotic isolating mechanisms in these studies. So to address this, I have data documenting reproductive isolation in 8 different ways that span both intrinsic, extrinsic, prezygotic, and postzygotic mechanisms. So I calculated reproductive isolation using Sobel and Chen's suggested equation here. And for this level of, or for this measurement of reproductive isolation, I just want to say that positive values represent conspecific, purebreds, or assortative mating, while negative values represent heterospecific, hybrid, or disassortative mating. So the pattern that I found is that we see overall snail eaters have much stronger levels of reproductive isolation than generalists. And this is primarily due to the fact that we observed 0 hybrid mating events with snail eater females in the wild. However, if we exclude those early barriers, we still see that snail eater females have stronger mate preferences in the lab, and that purebred fry have higher survival than snail eating hybrids. Lastly, I think this pattern shows that extrinsic barriers, such as wild courting success, mate preference, and even things like wild survival here at the end, seem to increase reproductive isolation, while intrinsic mechanisms seem to favor hybrid matings. So my main takeaway from all 3 of these studies is that we find a positive relationship between novelty and reproductive isolation, where snail eaters are more aggressive and had higher levels of reproductive isolation than generalists, but that this relationship seems to be maintained primarily by early prezygotic extrinsic isolating mechanisms and not due to low hybrid feeding performance. And with that, I'll say thank you for letting me share my research with you. Thank you to my lab for their support, and I'm happy to take any questions.